Hello friends, welcome back to my channel BioFrick. Today I'll be talking about different plant pigments. Okay, so in last class of plant physiology I had briefly mentioned different pigments, but then did we really look at the biochemistry of those pigments? So this is a good mix of plant physiology and biochemistry. So let's get started. So the first molecule that I'll be talking about is chlorophyll. Okay, now this chlorophyll is green in color. I told you that it absorbs most of the blue and red light, and it reflects green light, and hence its color is green. But then how the structure is given for chlorophyll? Okay, so this chlorophyll which is here, it is lipid soluble molecule. Okay, and that is present in thylakoid membrane. Okay, so since it is lipid soluble, it can easily stay in the thylakoid membrane. Okay, now this is the image of chlorophyll molecule, and there can be different types of chlorophyll, like there can be chlorophyll A, chlorophyll B, or there can be chlorophyll C. Okay, now remember that chlorophyll, it can either be something called as chlorine, okay, or there can be something called as porphyrin. What is this chlorine and porphyrin? I'm referring to the ring structure. Okay, so this is the ring that I'm referring to. Now when will I say that this particular chlorophyll is made up of chlorine? Okay, this is not that Cl chlorine. Okay, this is the structure, okay, of this chlorophyll. So when will I say that it is made up of chlorine? When there are 3 pyrrole rings, okay, and 1 pyrrolene ring. Okay, so now when there are 3 pyrrole rings and 1 pyrrolene ring, it results in chlorine structure. However, when we talk about this porphyrin structure has all 4 pyrrole ring only. Okay, and there is no pyrrolene. Okay, you might get confused, but then I'll simplify this further, that a chlorine is a large heterocyclic aromatic ring consisting of 3 pyrroles and 1 pyrrolene linked through this particular bond. Now where is this pyrrole in this whole structure? Can you see this? This is a pyrrole. This is a pyrrole ring. This is a pyrrole ring. So in short, what I can say is that further, if you look at pyrrole is made up of overall 5 atoms. Okay, out of which 4 are carbon and 1 is nitrogen atom. Okay, so there are 4 carbon atoms and 1 nitrogen atom. Total 5 atoms are there. So can you see that there is 1 carbon here, 1 carbon here, 1 carbon here, 1 carbon here, and 1 nitrogen. Now this is seen in this all structures here, isn't it? Now when I say it is connected by CH2 bond here and 1 bond here, this is something called as methane. Okay, this is a methane linkage. Okay, where can you see the methane linkage here? See, this is nothing else but CH double bond 1. So isn't it methane linkage there? So that is methane linkage. So I hope chlorine here is clear to you. So chlorophylls, like your chlorophyll A, B, and chlorophyll D, all these are chlorine derivatives. However, when I say chlorophyll C is porphyrin derivative. What do I mean by porphyrin? Porphyrin has 4 pyrrole rings. Okay, now basically it's reduction of 1 pyrrole double bond in porphyrin ring. Okay, so whenever you reduce 1 pyrrole ring, what it will result into is chlorine. But for this particular chlorophyll C, it has all 4 pyrrole rings. Now what I can say is that the chemical compound that contains 4 pyrrole or pyrrole like rings, they call as tetrapyrrole structure. So I can say that this is tetrapyrrole. Not necessarily all are pyrrole, but pyrrole like rings are there, right? So another interesting thing about this particular chlorophyll is that it has magnesium in its center, isn't it? So in last class as well I told you, like in hemoglobin we have iron, here that is magnesium. Now can you see the bonds there? So this is your magnesium, and it is connected to 2 nitrogen atoms with a covalent bond. However, the other 2 nitrogen atoms, that particular bond there is a coordinate bond here, isn't it? There is arrow like this, so that's a coordinate bond. So just remember that, another simple thing there. Then can you see this structure which is extra here? And this is again a ring, isn't it? It is a ring. And if you look at the carbon number, there are 5 carbons, so I can say that it is cyclopentanone ring. Why pentanone? Because there is a ketone group there, isn't it? So there is a cyclopentanone ring there. Okay, now this cyclopentanone ring is attached to the third pyrrole ring here. Okay, so it is named like this: 1, 2, 3 and 4. Now there is this fourth pyrrole, isn't it? There is this fourth pyrrole ring there. Now this fourth pyrrole ring is attached to a phytol tail there, and phytol tail that I'm talking about, it is hydrophobic in nature. Okay, it is hydrophobic in nature, and you can see that it's a kind of hydrocarbon tail. All are hydrocarbons here, and it is attached to the porphyrin or chlorine derivative ring, or to be more generalize this term, to more generalize this term, I can say that it's a tetrapyrrole ring. So how your phytol tail is attached to the tetrapyrrole ring is via the ester bond. Okay, now that is attached to the ring via ester bond, and there is also a propanoic acid group here. Okay, so that also should be taken into consideration. Now this phytol that I'm talking about, it is made up of isoprenoid. I mentioned this in the last class in brief actually. So to make it more clear, okay, isoprenoids are made up of 5 carbons. Okay, they are made up of 5 carbons, and there are 4 like this, that will make up 20 carbon long phytol tail. Okay, now there was one more thing that I want you guys to understand. It's very important, is R2 group that is present on second pyrrole ring. This R2 group is differentiating factor. You can basically differentiate between your chlorophyll A and chlorophyll B. Chlorophyll A has methyl group there, and chlorophyll B has aldehyde group. Okay, so these are the basic things which one should remember. Just remember that if there are 4 pyrrole rings, it's porphyrin, while if there are 3 pyrrole and 1 pyrrolin, then it becomes a chlorine derivative, and this chlorine derivative means basically ABD is a chlorine derivative, while your chlorophyll C is porphyrin derivative. Okay, and moving further, we need to focus on the chlorophyll A and chlorophyll B differences. What are the differences between them? So after learning the above part, you will understand that why there is a difference in this formula. Okay, how that difference actually occur, and it will help you to remember the formula. I don't think you should remember the formula, but then if you can remember, it is always good, isn't it? You know the reasons behind everything, every little small thing. So if you go to see, in chlorophyll A, as I said, there is extra methyl group. Here, what is there? In chlorophyll B, there is aldehyde group, isn't it? Now, when I see there is a when I say that there is a methyl group, there is CH3 there. So H is 72 here. Okay, carbon will remain same, your nitrogen will remain same, then your magnesium ion which is there in the center, that remains same. What changes is hydrogen and oxygen. There will be one extra oxygen here because of the aldehyde group, and that is O6. Here there will be O5. Then there is hydrogen 72. Here there is hydrogen 70. Okay, so H is just 1 here, but here 1 plus 2, that is CH3, so 72. Okay, not really that important, but just understand why there is a difference in that formula. Moving ahead, as I said, okay, these differences were spoken about last time, but I'll just quickly brief upon that. So chlorophyll A is a pure in the pure state, it is blue green, while this one is olive green or somewhere yellowish green. Okay, chlorophyll B. Then moving ahead, chlorophyll A is essential photosynthetic pigment, while this one is accessory pigment. Then there are here the pyrrole ring 2 contains methyl group, and this pyrrole ring 2 contains aldehyde group, and chlorophyll A absorbs more red light, okay, than violet blue light, and because of that you can say that it's reflecting the blue light, because it's absorbing less of blue light, so it will reflect more of blue light, and it's blue green in color in its pure state. Here obviously it will be opposite, okay, to this olive green, yellowish green, so it absorbs less of red wavelength of light. Okay, now moving ahead, okay, we will be talking about another pigment here, that is carotenoid. Okay, before that there is something called as bacteriochlorophylls. Okay, and this bacteriochlorophylls which are there, okay, they can be of type A, B, C, D, and E. Okay, you can write them something like this, like CH chlorophyll A, then bacterio chlorophyll B, and so on. Okay. Now, these bacteriochlorophylls are obviously, as per the name, they are present in an oxygenic photosynthetic organisms, especially certain bacteria. Okay, and they absorb the light of longer wavelength. Okay, they absorb the light of a longer wavelength compared to the chlorophyll molecules, the general chlorophyll molecules that we saw. Talking about now the third type here, that is carotenoids. Okay, now in carotenoids, these are said to be the scavengers. They act as scavengers, or they basically protect the plants from harmful radiations. Okay, because I'll talk more about this later, but then let's first talk about the structure in general. Okay, so the carotenoids which are there, okay, they are made up of terpenoids. Okay, they are made up of terpenoids, and there are about 40 carbon terpenoid compound. Now 40 carbon terpenoid compound, the subunit, okay, of this terpenoid, isoprenoid, is made up of 5 carbon. So like this, 8 subunits come together and they form 40 carbon terpenoid compound. Okay, these terpenoids and all are important in the secondary metabolites as well, which I'll cover later. For time being, just remember that even the chlorophyll phytol tail has something called as terpenoids. They were made up of tetrapenoids there. Here it is 40 carbon terpenoids, not necessarily always C40, but generally you will find C40 there. Okay. Now this whole carotenoids, okay, which are lipid soluble, again these are also lipid soluble, they are subdivided into 2 classes. Okay, so the 2 classes are something like this. Class 1 is beta carotene, which is there in carrots, and you have xanthophylls, also known as lutein. Okay, so have you seen in autumn, okay, the leaves of the plant, they turn oranges, or not oranges, yellowish there. This is because xanthophyll pigment is because of xanthophyll pigmentation, you can say. Okay, so the wavelength, okay, the absorption spectrum of this carotenoids is somewhere around 400 to 500 nanometers. Okay, now moving ahead, I want you guys to know how these carotenoids are acting as accessory pigments, and they are acting as something called as quencher. Okay, I don't know if you are familiar with the term quencher here. Okay, they are quenchers, and they also act as scavengers. Okay, so here what do they do is that they protect the plants, okay, against certain radiation. Like for example, let's say a particular chlorophyll molecule which is there in a photosystem is getting excited, and it is getting so much excited that it's not returning directly to the ground state and it's taking too long. Then it goes into the triplet state, and here something called as photochemical quenching occurs. Okay, basically the singlet oxygen is here released. Okay, this particular oxygen that is formed here, it is very much harmful to the plant. Okay, it can damage the cellular components. Okay, the extremely reactive singlet oxygen damages the cellular components. Now this particular phenomenon, what all happens is that the photoproducts here in this whole procedure, wherein the radiations are exciting this chlorophyll, and then it's not returning to the ground state, and there is singlet oxygen is getting formed. There can also be something called as photo inhibition. Okay, there can also be damage to D1 protein here. D1 protein is a part of photosystem 2, and this particular phenomenon can also damage your D1 protein of photosystem 2. Now carotenoid here enters like a hero. Okay, now this carotenoid exerts their photoprotective action by rapidly quenching the excited state of chlorophyll. Okay, so it will rapidly take in all that excited state on itself now. Okay, that's how you can remember. Okay, and now this is something called as non-photochemical quenching. Okay, it is something called as, see, before there was photochemical quenching. Okay, before there was photochemical quenching, it was damaging the plant tissues or plant cells. Okay, as I said, it was interfering with the overall photosynthesis process. It was inactivating your D1 protein. But when your carotenoid enters, okay, it does something called as non-photochemical quenching. Okay, now how does it do that? Okay, basically what is happening is photochemical quenching is the quenching of excitation energy by a process other than photochemistry. This was a photochemistry wala process. Okay. Now in this case, carotenoids accept the energy from the excited chlorophyll. From here, the carotenoid now will accept that excess energy, okay, and dissipate that energy as heat. So here what will be released is heat. Rather than damaging those cells, the excess energy will be dissipated as heat. This dissipated excess light energy is achieved by something called as xanthophyll cycle. Okay, so there is something called as xanthophyll cycle that occurs. Okay, where you have your, sorry for that, here we have something called as viola xanthine, okay, or viola xanthine you can say. So that viola xanthine is undergoing a process called as deepoxidation. Okay, it is undergoing something called as deepoxidation, and it is getting converted to anthraxanthine. Anthraxanthine is getting converted to zeaxanthine. Okay, so this is the process when there is high intensity light. Okay, so this is the conversion that will occur, okay, in one case, in one scenario. Okay, if this defense is not sufficient and toxic photoproduct forms, even in spite of doing this xanthophyll cycle, carotenoids eliminate the reactive photoproducts by acting as scavenger. So that's why we term them as scavenger. Basically they are protecting the plant. Okay, now moving further, we have something called as phycobilins. Okay, how many pigments did we see? We saw 3, and this one is the 4th one. Okay, so there is something called as phycobilin here. Now these phycobilins which are there, these are non-cyclic linear, okay, tetrapyrols structure. Okay, so these are non-cyclic at first place, you should remember this, and they are linear, not cyclic. Linear tetra meaning 4 pyrols. Again we are talking about pyrols. Okay, pyrols means 4 carbon and 1 nitrogen. Overall 5 atoms are there, and we can easily see, if you look at this 2 structure randomly, you can see that there are 4 pyrols. Again, it's a linear chain, unlike your previous one which were circular sometime, okay, that chlorophyll ring and all. So here now, another important thing about them is that this is similar to bilirubin. Have you heard the term bilirubin? Okay, bilirubin is seen in humans. It accumulates, and actually there is a metabolic pathway for metabolic breakdown of this bilirubin. When it accumulates over a certain range, a person gets jaundice. Okay, or this is one of the symptoms, like if it's accumulating, something is wrong with your liver or that metabolism. So now the point here is that this bilirubin structure and this phycobilin structure is similar. Okay, and here another thing that you should remember is that these are not lipid soluble, they are water soluble. Okay, all these are water soluble. Okay, now since they are water soluble, they will solubilize in water, right? They won't be present in the membranes. As I said, the carotenoids, they were there in thylakoid membrane because they were hydrophobic in nature. Are these hydrophobic now? These are hydrophilic. So what do they do is that they form something called as phycobilisome. They adhere to the membrane, and this is that phycobilisome. It's a membrane here. Okay, now they act as accessory light harvesting pigments. Okay, the phycobilins, they act as accessory light harvesting pigments which capture the light energy and feed it to the chlorophyll molecule present in the photochemical reaction center. Okay, now the next thing that we will be looking at is the presence of all these pigments, how they are distributed throughout the biodiversity here. Okay, all these different plants or prokaryotes. So just remember that your chlorophylls which are there, what is important here, I'll just display, you can take a screenshot of this and remember. But then what is important here is that the cyanobacteria which is here, cyanobacteria has more amount of chlorophyll A, and it also has carotenoids and phycobilin. And another important one where phycobilin is seen is red algae. So phycobilins are present in red algae. Otherwise this table is also important, you guys can take a screenshot here. Then what about the locations of these pigments? Okay, so in oxygenic photosynthetic organisms, the eukaryotic photosynthetic organism here, the first one, it is present in the thylakoid membrane of chloroplast. Again, the prokaryotic photosynthetic organism present in thylakoid membrane, and anoxygenic, talking about the green photosynthetic bacteria, it is present in a place called as chlorosome. Okay, it forms something called as chlorosome, and it is also seen in the plasma membrane, while your purple photosynthetic bacteria is present in the chromatophore. Okay, it is present in the chromatophore of the plasma membrane. So this were the exceptions or something new about these two bacteria, so that can be remembered. So that was it, guys. See you in the next lecture.